Welcome to STEMpaths. I'm Dr. Thornton Larson, and this is STEMpaths, where students unpack papers, study paths, and the people behind them. Today, our guest hosts are Victoria and Daisy, and they're going to be interviewing our guest, Mr. Richard Orton, from SMU up in Halifax, Nova Scotia, of all places, where he's currently studying mostly genomics of whales and things, which is interesting based on when we first met and crossed paths. But with that, I'm going to let you guys take it away. So take it away.
Well, hello, Mr. Richard. Just for a brief formal introduction, my name is Daisy Barraza. I'm a senior here at Sora State University, and I'm doing the pre-med route for hopes to become an anesthesiologist.
Okay, cool.
Hello, Mr. Richard. My name is Victoria Aguayo, and I'm a senior here at SORAS. I'm majoring in biology with a concentration in pre-health, and I'm looking to go to medical school to become a pediatric doctor. So
Nice.
Thank you for joining us. We're going to be talking about your research article titled Genomic Evidence for the Purging of Deleterious Genetic Variations in the Endangered North Atlantic Right Whale. After reading your research article, Daisy and I concluded that your team, including yourself, sole focus was on the genetics of the North Atlantic right whales and how those genetics may be affecting their population recovery. To start off, can you briefly explain what the study is about and what inspired you to investigate the genetics of North Atlantic right whales?
Yeah, so whaling globally had significant impacts on many different species of cetacean. In the 80s, there were international laws that were put into place that curbed whaling across many places in the globe. And since then, since whaling at least was slowed, we see recovery for a lot of species and a lot of populations that were potentially even on the brink of extinction. One example of a species that shows strong signs of recovery is the southern right whale. But there are also examples like the North Atlantic right whale, where despite international laws against whaling, we don't see strong signal of population recovery. So the impetus for this paper was to understand why some species like the southern right whale are recovering post-whaling and why other species like the North Atlantic right whale are not recovering following the seizure of whaling. And then specifically to understand if there was a genetic basis for the lack of recovery.
And for listeners who may not know why North Atlantic whales are declining, what is their lack of population recovery so concerning since the 80s?
It's not that they're still in decline. There's a lot of science that goes into it to understand if they are declining, if they are slowly recovering, or if they're just kind of treading water, no pun intended. Whales are they're not like whales are not terrestrial, and therefore it's difficult to estimate the population size. I mean, it's difficult with terrestrial species too, but with whales, it's hard to even detect them. They spend most of their time under the surface of the water. Right. So there are a number of different models that are updated every year to try to understand how many calves are born each year and then how many whales are actually lost each year. Most of the North Atlantic right whales that are lost to anthropogenic causes are through ship strike and non-lethal entanglement. And then the reproductive rate yearly, there's usually somewhere between, 5 and 20 North Atlantic right whale calves that are actually detected. But we also don't know how many calves are born and then maybe die early in life before detection. So we don't see like the population growth that we see for the southern right whale and then other species, maybe like populations of fin whale.
Okay. So the overall main goal of the research was to kind of give modern research a reason to unfold more discoveries about population, or can you give us a little more intel on that?
Yeah, so part of the one reason that the North Atlantic right whale could be failing to recover at the rate of other species like the southern right whale is because of maybe a lack of genetic diversity or inbreeding depression in particular. So the idea was to understand if there is ultimately a genetic basis for the lack of recovery.
Okay.
Your paper states that North Atlantic right whales show extremely low genetic diversity compared to southern right whales and bowhead whales. Can you explain why their genetic diversity is so low and what the consequences of that are?
Yeah, so it appears there's another paper that unraveled the demographic history of the southern right whale and the North Atlantic right whale. And it looks like for most of the history of the species, the North Atlantic right whale has been a smaller population. The numbers have been less compared to the southern right whale, which has high genetic diversity. And there's a number of reasons why the southern right whale could have just been a larger, there could have been more southern right whales than North Atlantic right whales. But we see in the demographic history, you can see the impacts of whaling for both species. So potentially whaling reduced the numbers of North Atlantic right whales, and then this drove down genetic diversity. When populations get small enough, genetic drift increases, which reduces genetic diversity within a population. But also smaller populations have higher rates of inbreeding, which further reduces genetic diversity. So we call this the mutation vortex or the extinction vortex.
Okay. You also
And that's
found,
likely. Go ahead.
I'm sorry.
Go ahead.
You also found strong signs of historical inbreeding in North Atlantic right whales shown through high inbreeding coefficients and long runs of homozygosity. How does this long-term inbreeding shape the population today? Also similar to the mutations that you were speaking of.
Yeah, so the runs of homozygosity are they're an estimate essentially of what inbreeding is like in the population. An individual who is the result of a full sibling-sibling relationship could have runs of homozygosity that span most of the genome. And this is important because deleterious mutations they can segregate in any population, and they do segregate in most populations or all populations. But deleterious mutations also tend to be recessive. So if you have a large population that has a lot of genetic diversity and high heterozygosity, that heterozygosity can shelter those harmful mutations in heterozygous genotypes. And when populations get small and inbreeding increases and homozygosity goes up, those deleterious mutations get exposed in those runs of homozygosity. And when those recessive deleterious mutations, where they're found in homozygous genotypes, they have to be expressed. That whatever phenotype is linked to that deleterious mutation gets expressed.
Your team identified 41 genetic variants that were common in females who had never calved, but rare in females with high reproductive success. What do these specific variants suggest about reproduction or embryo development in this species?
Yeah, so I had mentioned earlier that marine mammals or aquatic species or other marine species, it's difficult to detect these individuals and these taxa. There's a population. So for comparison, there's a population of wolves. I think near Michigan, they might not exist anymore, but they did for a while. It was the Isle Royale wolf, and it was founded by a handful, maybe 2 individuals, I think in the 40s. And this population has kind of been like a natural experiment of inbreeding depression. And there have been several reintroductions of wolves to that island population to try to increase genetic diversity. And we've seen it. We've seen genetic diversity go up and then fall again several times. But we also have found, not me personally or my team, anyone I work with, but in general, scientists, researchers have found a high number of neonate mortalities on that island, where neonate mortalities in the ocean get lost and we often don't detect them.
Right.
But on a terrestrial environment, we can find those neonate mortalities. And those neonate mortalities in the Isle Royale wolf population, most of them had spinal abnormalities. And it was interesting that we found that the nulliparous female North Atlantic right whales carried mutations that were linked to spinal abnormalities. And then the females that we sampled who had been spotted with at least a few calves did not carry those mutations. So it's possible that we are seeing some sign of inbreeding depression in the North Atlantic right whale that's ultimately linked to spinal abnormalities. It's possible.
The paper explains that human-caused stress, such as vessel strikes and fishing gear entanglement, remain the main drivers of mortality. How do these human impacts interact with the genetic vulnerabilities you found?
Yeah, so there's someone else in the lab that I work in who is doing research in epigenetics. And epigenetics is essentially the methylation of the genome, or that's one facet of it anyway. And the methylation of genomes can be linked to stress. So when these whales are getting entangled in fishing gear from lobster fisheries, they don't die immediately. And a lot of times they're untangled and they live the rest of their lives. Most of the North Atlantic right whales have been entangled at least once. And in a lot of cases, whales have been entangled multiple times. And what my lab partner has found is that whales that have been entangled or have been entangled multiple times, their methylation patterns are different than whales who have only been entangled once or no times at all. And this is likely linked to stress. So what we're seeing is this genomic signal of the stress that's caused from being entangled.
Okay. Southern right whales and bowhead whales show signs of population recovery, while North Atlantic right whales do not. What genetic difference between these species might help explain why only some are recovering?
Yeah, it's still hard to know exactly why. The southern right whales in particular have high genetic diversity, but there are a lot of individuals still on the planet, and there are multiple populations of southern right whales, whereas the North Atlantic right whale, there's only one population in the entire species that exists. I think there's maybe 372, 380 North Atlantic right whales left. And don't quote me, but I want to say there's tens of thousands of southern right whales on the planet. So the census population size and genetic diversity are not always correlated, but often they're correlated. So bowhead, southern right whale, just larger numbers, the population, higher genetic diversity. And we don't know exactly, again, why that is. It appears, again, that the North Atlantic right whales probably just had a smaller population size throughout its history.
Your conclusion suggests that genetic data should be incorporated into future conservation strategies and population recovery models. What would using genetic data and conservation actually look like in practice?
Yeah, so one example of this is trying to understand what the recovery goals for the North Atlantic right whale might be. And we've used genetic data to understand what the historic population size might have been for the North Atlantic right whale. So we can use genetic diversity in that way to kind of set some sort of goal to aim for, as far as like what are the numbers that we're hoping to see. We can also do genetic monitoring, where maybe every year, just for example, we could sample the calves and we could estimate these runs of homozygosity every year across every generation. And what we would hope is that over time we see heterozygosity or genetic diversity increasing.
Okay.
I think that's all the questions we have, Dr. Larson.
Oh, on the paper?
Yeah.
Okay, well then let's transition a little bit into other aspects of it. So tell us a little bit of the writing process on putting together the paper, like how you led the charge on putting together most of the paper itself.
Yeah, when I came in, the data were already generated. So someone else, there's a huge team of people working with the North Atlantic right whale. There's a consortium every year, and I think maybe they allow 200 to 300 people every year to come to the consortium, but there are more who are working on this. So there are people who their job is to disentangle whales from fishing gear. And when it comes to North Atlantic right whales, they can take a sample of maybe blubber, just a little plug that the whale doesn't feel, and they can send that to someone like my advisor, who has graduate students like myself, who will extract the DNA. There's a huge sample or tissue collection that my advisor has, and another graduate student, and my advisor sat down and decided which samples, which whales to actually sequence their genomes. That was all done before I got there. So when I came in, what I had were essentially sequences on a computer. And that's where I started the bioinformatics process of mapping those reads to a reference genome and then filtering those data.
Cool. So I know also you've communicated results in conferences. You've done it in positions where you've done it with both lay audiences as well as very knowledgeable audiences. What's your favorite way to communicate research findings in both situations?
Yeah, I am learning to communicate with the general public more and more. I think it's going to become increasingly important for me to do that as I transition out of graduate school. I think so far I've enjoyed giving talks to maybe professors and other graduate students and postdocs because I can get valuable feedback. And it opens the door for discussion where in the past, like I've kind of come at this from a perspective of an interest in evolution. And I came into a conservation genomics lab with this interest in evolution. And I've kind of merged the two. But over time, I've recognized the importance of conservation, and it's become more important to me personally. So I think I'm kind of transitioning away from evolutionary biology to like the application of it in conservation genomics.
Very cool. Very cool. What is the application of more of that conservation versus more of a phylogenomics approach look like?
Say that again.
The using genomic resources to answer conservation questions versus genomic resources just to look at relatability through like a more of a phyllo process.
Yeah. I mean, I think conservation genomics is essentially the application of evolutionary genomics or evolutionary biology. Maybe not always, and maybe there's a little bit of a lag where questions are asked in the lab or just behind a computer and data are generated or simulated, and theory is kind of born and tested with simulated data. And maybe over time, as accessibility to genomic resources increases, people can go out and then kind of test theory in natural settings with empirical data. And then I think as more time lapses, we start learning how to actually apply those sorts of methods and those questions to trying to conserve biodiversity. Mutation load, the idea has probably been around since before the 50s, but the term was first coined in the 50s and it was applied to the human population. So you two are talking both about going into medicine. I feel like mutation load is something that will be adjacent to your work, no matter what it is that you do, if not like at the forefront. Genetic diseases, I think they say that most humans carry maybe 5 deleterious mutations in their genomes and our genomes, that if we were homozygous for those mutations, they would be lethal. So those harmful mutations are segregating in populations, in the human population. And I think over time, this is just an example, we've kind of learned to take that theory, because at first it was a theoretical paper, and then there were theoretical genomics or genetics done around it, and then applied to other populations like, I don't know, ibex or whales or pigeons or whatever. And now we're kind of learning how to apply it in a conservation sense. I read a paper earlier today in birds in Africa where they had museum specimens across a time period, and they estimated mutation load across that time period, and they saw that in these generalist species of bird in Africa, that mutation load is actually decreased over time despite anthrogenetic disturbance. So that's one way that theory has kind of been ultimately applied to conservation.
Cool. So, I mean, I know we've talked that getting into the cetacean side of thing is fairly new, but what has been the most rewarding aspect regarding your journey in science in general?
Yeah, I don't know if I'm quite yet there.
Well, so far, it's
Actually,
a very so
yeah.
far.
I'll back up. I'll kind of start in the future and kind of back up to the present, and then maybe the past. Yeah, I'm looking forward to having my PhD. So hopefully that's something that does happen. I'm not there yet. I've not crossed that finish line. I think that if that happens, that will be rewarding. And I think going into, well, whatever it is, I'm not 100% sure I want to do conservation genomics, but let's say I do go into conservation genomics, I could see that being a rewarding field. Um, I think so far, though, to date, we're still in the process of figuring out exactly how we want to apply the genetic data and the answers that we've gotten in the lab that I'm working in. So ultimately, I'm working off of a grant that has two parts to it. There's the genetic component, and then there's a sociology component. And the sociology component, the goal is to figure out how to take the findings from the genomic research and to communicate that to a broader audience. And what we're learning is that the message will probably have to shift slightly differently for communicating to the general public, for communicating those results to the fishing industry, and then communicating those results to policy implementers and managers. Because it's important, I think, if we're going to preserve the North Atlantic right whale, it's important that minds are changed at the policy level, at the fishing industry level, and in the market. And those things aren't exclusive of one another. It all works together, but I think communicating the science to those three groups will be slightly different. And we're working to figure out how to do that. And I think me sitting in the back of the room, for the most part, and listening to the managers and the policymakers kind of discuss how to do this, and listening to the sociologists discuss how to best to do this, is really interesting and it's rewarding.
Cool. As far as your journey goes into these positions, like, when did you first have the epiphany that going into research as a career was something that you wanted to pursue? When was it that you were like, man, I want to do this and I can do this?
Yeah, so it started out just with a fascination of reptiles, and in particular, a fascination with venomous snakes. And I had a small collection of reptiles that I kept at home as a hobby. There are surprisingly a lot of people who do this, especially in Texas. And I think one day I realized that I could do more than just keep snakes and try to keep them happy and to breed these snakes. I realized that people were doing like scientific research with venomous snakes. In particular, there were people with big personalities doing venom research. And that kind of set the ball rolling. And I found my way into a lab at Georgia Southern University that did not work with snakes, but worked with Florida scrub lizards. And then in that lab, I was taught more broadly how to do science, and then I kind of slowly transitioned into doing population genetics while I was in that lab it was a way of starting to learn how to do molecular biology and to learn molecular biology techniques.
Awesome. So we're going to run into our wrap-up questions here, which are just 3 questions we ask all our guests, and I'll have the ladies ask you those 3.
What's one piece of advice you'd give your undergrad self?
My undergrad self, yeah. I was a terrible undergraduate student. I was the worst undergraduate student that I think I can think of. Yeah, every now and then I think I have a student who's late to class all the time and is hungover half the time. And yeah, I think if I could go back, I would tell myself to get it together a little bit, to grow up, and to maybe pace myself a little bit more. But if I had not done those things, I don't know, I might not be here.
Yeah.
I wouldn't. I would be somewhere else, which could be equally good or better. I don't know. It's hard to say. Yeah, I mean, I've made so many mistakes, but at the same time, I kind of don't necessarily regret those mistakes.
Mm-hmm.
So yeah, that's a tough loaded question. Yeah, I could give advice to other people who are working in their undergrad, and maybe that would be to ask yourself, and it's going to change, but I would tell people, ask yourself, what is it that you actually want to do?
Yeah.
And to maybe think about the day-to-day. If you're going into medicine, what is your day-to-day actually going to look like? What's the stress load going to look like? I think some people just have a natural passion for things. And if you do, I say follow that. Just go for it.
Mm-hmm.
I'm also the last person who should give anyone advice. But yeah, I believe when you really want to do something, like you should go for it and see what happens and give it your all. But for those people who kind of are not sure what they want to do, so I wasn't sure what I wanted to do wrapping up my undergrad when I slowly became a little bit better of a student. I did somehow miraculously find a way to graduate. I think at that point I had no idea what I wanted to do. And sitting and thinking about it probably would have gotten me where I was going a little bit quicker. Mm-hmm. It's similar advice that I was given in graduate school to what questions was I interested in actually asking and pursuing? Because, that's what science is. You're pursuing questions, at least in the theoretical side. I guess when it comes to applied sciences, like medicine, you're actually applying and you're doing.
Yeah. If you weren't doing this, what would you be doing?
I have no idea. I can tell you that right now, as I'm approaching the defense of my PhD, I'm trying to figure out what I want to do. And I don't think academia is for me for several reasons. So to me, it's kind of the two options that I see that are glaring is pursuing conservation genomics or going into bioinformatics. I was offered a job at one point working at a hospital in Houston as a bioinformatician. I would have been working with a team of other people who had more experience than I did. So I would have received a lot of training, but the idea would have been working on a pipeline to analyze human genomes to detect signs of cancer or other genomic diseases. So there are jobs like that. So I think right now the two things I'm mostly considering would be bioinformatics and probably a medical, in the medical field broadly, or conservation genomics.
And last question, what's one thing you wish more people understood about science or scientists in general?
Yeah, I wish to God people understood that, when we do science, we approach things with the best data that we have to date. We approach things with the best known science or the best science to date. And maybe the speed of light is the speed of light is the speed of light. But certainly when it comes to biology, when it comes to medicine, when it comes to disease, there's a lot of variation. It's incredibly complex and things change constantly. Biology, the world itself is dynamic, and living organisms are dynamic by nature. Genomes are constantly evolving. This is just how things are. And I wish people would understand that science is a process and it's our best answer at the time. And it's incredibly difficult. And I think there are a lot of people right now who are just turning their back on science because they've realized that science isn't perfect. And that's I don't know, what are you turn your back on science, and then what? You've got nothing. You're operating without any kind of logic or reason. Mm-hmm. So yeah, I wish the general public understood that a little bit more.
All right. I want to thank our guest today, Richard Orton, for making the time to come and talk to us. It's been absolutely fantastic. I want to thank our guest hosts, Victoria and Daisy, for coming in and asking Richard all those questions, because I think it was a great time. I'm Dr. Thornton Larson, and this has been STEMpaths. So thank you all for coming in, and we'll see you next time.